All right, I think we can go ahead and get started. Again, thank you for everyone for attending today. We're delighted to have Dr. Mike Dudley here to present his talk titled "It Takes Two to Tango: PK/PD Considerations for Beta-lactamase Inhibitor Combinations for Drug-Resistant Pathogens." So again, thank you to Dr. Dudley for agreeing to attend today. Today's session is going to be recorded, and a recording will be made available after the seminar. I'll more thoroughly introduce Dr. Dudley in a few minutes, but I'd like to just briefly introduce myself and my co-moderator, Dr. Gauri Rao. My name is Zack Bulman. I'm an assistant professor at the College of Pharmacy in the Department of Pharmacy Practice at the University of Illinois at Chicago. Gauri, do you want to introduce yourself as well?
Yeah, thank you, Zach. And again, thank you, Dr. Dudley, for agreeing to be a part of this session. And again, I'm Gauri Rao. I'm an assistant professor at the Eshelman School of Pharmacy at the University of North Carolina.
Excellent, thank you. We'd also like to thank our organizing committee for today's webinar. In addition to Dr. Rao and myself, also Dr. Jian Li and Dr. Jaime Laura Tamayo, as well as some of our trainees and junior colleagues who are all shown here. So we would greatly appreciate everyone here's help and assistance and coordination for today's webinar. As far as disclosures, Dr. Rao and myself have no conflicts of interest to report, and there was no funding for this seminar, and Dr. Dudley will mention his just in a few moments. Just a few quick housekeeping items before we get started. If you would like to ask a question, we do have the Q&A feature enabled for today's session, so please go ahead and get those typed in during the meeting. Today's seminar will be approximately 40 minutes of slides presented by Dr. Dudley, followed by a 15-minute Q&A session, and we'll do our best to try to get as many of those questions answered as possible at the end. So now to properly introduce Dr. Mike Dudley. Again, we're so grateful, and it's an absolute pleasure to welcome him today. Dr. Dudley is the president and CEO of QPEX Biopharma. He has an extremely impressive resume. I'll try to just highlight some of the key points here. Has over 30 years of experience in anti-infective drug research, development, and commercialization. He has overseen the development and regulatory approval of multiple compounds, including a beta-lactamase inhibitor combination in a new chemical class, which I'm sure many of you are familiar with, vaborbactam. Just to take a quick step back, he has completed his undergrad at Pepperdine University, did his Doctor of Pharmacy at the University of California, San Francisco, also did his residency there. He also did, and then went on to do his fellowship in infectious diseases at Hartford Hospital, and worked in academia and was successful as a researcher, studying PK/PD of several anti-infective agents. Became a professor of pharmacy and chair at the University of Rhode Island College of Pharmacy, and was also an adjunct professor at the Brown University School of Medicine. So after his tenure in academia, he worked for several biotech companies, then became the co-founder and chief scientific officer of Rempex Pharmaceuticals, before co-founding QPEX, where he's currently the president and CEO. Dr. Dudley has published over 150 articles and book chapters in his scientific career. And so without further ado, I'd like to welcome Dr. Dudley, and he'll share his slides and give his presentation.
Well, great, Zach and Gauri, and like to thank you as well as the organizing committee for asking me to talk about a topic that certainly I enjoy, both drug discovery as well as talking about resistance mechanisms and how we can overcome them using beta-lactamase inhibitor combinations. So let me get my slides up here and I'll get us going here. So hopefully that's all showing up well for everyone. So good. So let me just say, as well as maybe obvious, that I'm an employee of QPEX Biopharma. We're developing some of the agents that are to be discussed today. We have no commercial products to discuss with you. I guess that's good news, bad news, right? But some of the drugs I am going to, and uses that I am going to be talking about today may be investigational and have not been approved by any relevant regulatory authority in your jurisdiction. I know we have people who are from around the world here. but those regulatory authorities have not reviewed or approved some of the drugs as well as uses that I may be discussing today. Let me just tell you, while I'm giving you my disclosure, a little bit about QPEX. You may not have heard about it. QPEX is a small biopharm company that's based in San Diego. We spun out from the medicines company, meaning that when the medicines company was divesting itself of the commercial products and the entire infectious disease business, we formed QPEX and took the preclinical programs, which I'll be talking to you about, one of the members of that, into QPEX, and raised a venture round to support the company. QPEX has an experienced team of drug development scientists. I have been privileged to work with over anywhere from the last 15 to 20 years and we've been successful in bringing forth in the last decade, in fact, 4 regulatory approvals for drugs, one of which that Zack kindly mentioned being vaborbactam. We have a lot of partnerships that we have. We're known as public-private partnerships. Most significantly has been a BARDA partnership that we have now. That is a unique partnership that's advancing a portfolio of antibiotics that I'll show you in just a second about deep into development. That really allows us to be very thoughtful about both what that portfolio contains, but also in terms of how we do it. And we've also been involved in drug discovery work funded by the NIH, and we're actually with one of the co-chairs here with Dr. Jian Li at Monash University that resulted in the discovery of a new polymyxin with improved pharmacological properties. And as I'll talk about in a moment with particularly one of our programs involving beta-lactamase inhibitor products, we're really focused on those that CDC and WHO considers pathogens that are for which there is a critical need for new drugs and new therapies, and primarily those being Acinetobacter and Pseudomonas and Enterobacteriales. But we're also mindful that many of these infections are also occurring in the outpatient setting, and so are developing both IV and oral products for those as well. And this is a snapshot of how this portfolio is designed. We had the ability with our partnership with BARDA to really think very depthfully about kind of the end-to-end experience or patient type that you see. You're starting on the left side with many of these infections occurring in the outpatient center setting. And then, of course, when we deal with patients in the inpatient setting, many of those become critically ill with some of the most difficult to treat infections, such as those due to Pseudomonas and Acinetobacter. And our portfolio kind of lays on top of those settings there. It's anchored by a beta-lactamase inhibitor that I will tell you about that's currently investigational and working through phase 1 clinical trials. It can be delivered both orally as well as IV, and hence be paired with oral or IV beta-lactam antibiotics to address those patient settings. And then on the far right, you can see there that again, for those most resistant infections due to multi-drug resistant Pseudomonas and Acinetobacter, where oftentimes one may be using even combination therapy. We have a polymyxin, synthetic polymyxin, that's also in phase 1 clinical trials. So where I'm going to go today is I'm going to talk about new beta-lactamase inhibitors and why would it seem that we would maybe need to have another one, given some of the more recent drug approvals in this area. I also want to talk about antibiotic resistance and mechanisms of resistance and drug discovery. And I think that will help set up, the overall way that we think about this problem in developing new drugs. But then most importantly, I think the PK/PD consideration for beta-lactamase inhibitor and combinations, and some of the PD concepts that are helpful to think about, both from the idea of being able to evaluate these compounds once they're approved, but also from the standpoint of how we think about drug development. What are the relevant PK/PD indices that may be useful for these drugs in terms of the development and evaluation, and then importantly, the dosage regimen design. And so what I'll do here is I'm going to go through these 4 points, I think, and it will help you understand kind of the roadmap that we put ourselves on when we're discovering and developing these drugs. And one is first of all, you sort of start with what do you need to do in terms of a beta-lactamase inhibitor with respect to improved properties that can overcome class-based resistance, that is resistance that is relevant to the beta-lactam antibiotics. But then I'll also talk about what are known as general intrinsic resistance mechanisms, which are really the multi-drug resistant mechanisms. So when you're talking about multi-drug resistance, you're talking about, in general, the general intrinsic resistance mechanisms, such as those involving permeability as well as efflux. And I'd like to give a shout out in terms of setting this up. If you have access to wherever you get your favorite podcast, the Society of Infectious Disease pharmacists recently dropped a podcast chaired by Dr. Aaron McCreary with Dr. Ryan Shields and Dr. Robert Bonomo, talking about resistance mechanisms and particularly focusing on beta-lactam antibiotics and those resistance mechanisms. And I highly recommend that podcast to you all as kind of an additional primer on understanding these mechanisms and how to evaluate drugs for use in your patients, as well as just how well the products perform. The second point then is I'll talk further about some of the concentration response relationships for beta-lactamase inhibitors and how one might be able to express that and understand that in terms of dosage regimen design. And then finish up really on PK/PD issues of identifying then what the target PK/PD indice for inhibitory activity for beta-lactamase inhibitors that we use in preclinical models of infections with various companion beta-lactam antibiotics. And then I'll give an example from the meropenem vaborbactam program that shows how one can integrate then these properties and using data from both phase 1 and phase 3 trials to really help get the right dose into the patients that actually are going to need these drugs. So why develop a new beta-lactamase inhibitor? Despite the fact that these agents have been around now for a couple of decades, there is still a lot of problems left on the table that we sought to really improve in our discovery program. One is to be able to do a better job at pathogen coverage, and pathogen coverage with various inhibitor combinations has been spotty. In the best of situations, you're able to get at least 2, that being the Enterobacterales as well as Pseudomonas as part of that. But getting activity against Enterobacterales and Pseudomonas and Acinetobacter has really been an elusive concept until recently, which I'll show you that in a moment. The other is really getting then inhibiting all the major resistance enzymes, the beta-lactamases that are present within these bacteria. We've done a good job over the years of inhibiting the class A enzymes and some of the class Cs and class Ds, but the metalloenzymes, which are really very, very different because they have a different active sites, have been somewhat elusive until recently of being able to now find compounds that can actually bind in the active site of a class B enzyme and inhibit its activity. Now, a major part of the problem has been is that not having molecules that escape these MDR resistance mechanisms and permeability and efflux has been a problem. And that's part of the reason why pathogen coverage is oftentimes spotty. So for example, vaborbactam really has no important activity in Pseudomonas, largely because it's effluxed quite effectively in the efflux pumps in Pseudomonas, and therefore one is not able to really see much activity in Pseudomonas, even when you have an enzyme that should be inhibited. The other point is that we have to be mindful about emerging resistance, and in particular emerging avibactam resistance, a very unique resistance that takes place actually in the enzyme, the KPC enzyme. That's actually quite easy to select in the laboratory and has been problematic in patients, has been described by several groups where one can emerge resistance to avibactam. So thinking about a drug that would not be susceptible to these types of resistance mechanisms would be important. Now, we also kind of reimagine the idea about how beta-lactamase inhibitors, which have been traditionally given as fixed combination products in combination with beta-lactam antibiotics. But as we have been working in this area and really addressing those elements on the left-hand side of the slide, we have found that there really haven't found a beta-lactam that doesn't benefit by having a beta-lactamase inhibitor combined with it. That is, by being able to inhibit the enzyme and these in those bacteria, it just improves the activity of the partner beta-lactam, and also importantly can then minimize resistance that can take place as part of that. So in terms of how we reimagine this, is that there may be a situation in the future where in fact one could use a beta-lactamase inhibitor in a non-traditional sense, where it's not co-formulated as a fixed combination product, but would be available then for co-administration with various beta-lactam antibiotics, where you could in fact individualize the therapy in individual patients, much like what we do in other types of infectious diseases, but more in the virology area, or in fact, even if you go outside of infectious disease to the cancer area. And then lastly, we've thought about oral therapy. Many of these infections are now occurring in the outpatient setting. Fluoroquinolones, when I went into practice a long time ago, had just been approved, and that was a game changer in terms of practice within the institutional setting and the hospital, of being able to de-escalate patients from IV to oral therapy and be able to set them up for going home sooner. That, of course, has been lost with fluoroquinolones because of increasing resistance and, of course, improved recognition of the toxicity with those drugs that takes place. So being able to have something that would be able to then recover that ability to treat these infections in the outpatient center would be great. So here is how for the boronic acid beta-lactamase inhibitors, which Zack kindly mentioned earlier, which is a class of drugs that we, the first member of which, vaborbactam, that was innovated by our scientists that are now at QPEX, vaborbactam. The way that we think about then with innovation here is, as you know, is that rarely the first drug that comes from a new class of drugs is the best drug that's available. And when we developed vaborbactam and discovered that and got it approved in 2017, that compound actually went from discovery in our laboratories to FDA approval in under 8 years. And that was really accelerated by the early identification of the KPC enzyme, which threatened to really take off. So it was a compound that was really designed for KPC producing bacteria, didn't have very much important activity against some of the other carbapenemases at that time. And we continued to work in this area, and you can see the class 2 and class 2A and B agents there, which still relied on kind of this bicyclic structure involving a boronic acid with the amide bridge, which was able to then increase the activity of this class of drugs against class C as well as ESBL enzymes as well. And in fact, you could get a little oral bioavailability with this 73-23. But the next real breakthrough that took place is again, in these amide-linked molecules, was really being able to get molecules that then had metallo-beta-lactamase inhibitory activity. Now, was some, but not all, so good activity against NDM and some activity against the VIM, but no important activity against IMP was present. But certainly a breakthrough for this class. Another compound is currently in phase 3 clinical trials that I would characterize, taniborbactam, which is characterized by a similar structure and similar pharmacological activities. Taniborbactam would kind of fall into this class 2B group. But real breakthroughs then came in class 3, where then we were able to then broaden the metallo-beta-lactamase inhibitory activity, able to still get oral bioavailability now all using a prodrug. And we had switched then from the amide types of linkage to a sulfur linkage then to the heterocycle, which was really evident in terms of improving this activity. And then finally, our discovery efforts really culminated in QPX7728, where we really were able to make the molecule a little bit smaller, as you can see there. And instead of a fused 6-member ring, we have a cyclopropyl group that's appended off of there, which gave us really this ultra broad spectrum activity against metallos as well as serine beta-lactamases, and also now brought in activity against the oxa enzymes and ability to inhibit those in the Acinetobacter. And this was largely brought about by reduced effects on these multi-drug resistance mechanisms that QPX7728 is not as sensitive to compared to earlier members within this class. And of course, it also has oral bioavailability within rats. Now, before we get started here, I want to just clarify a few comments about multi-drug resistance. And there's a lot of terminology that gets a bit confusing sometimes in this area, with people talking about MDR and XDR and usual drug resistance and all of that kind of thing. And I find that those are sometimes not very helpful in terms of actually how to think about this area. And I'm just going to propose yet another way for you to think about this in terms of really thinking about multi-drug resistance being rooted in two general causes. One of those is class-based resistance. So beta-lactamases are a class-based resistance or modification of a penicillin binding protein, which is, of course, a class of drugs, being penicillins or beta-lactams or PAR-C, if you're thinking about fluoroquinolones. Those resistance mechanisms are class-based, and indeed, you can overcome them by making modifications on the molecules that can escape those. The other, though, broad category is the general intrinsic mechanisms, or general intrinsic MDR mechanisms that are referred to, is that those are involving permeability and efflux. And that's why you get multi-drug resistance that crosses classes. So you can have a pump overexpressed that can give you resistance to a fluoroquinolone, as well as give you resistance to a beta-lactam antibiotic, and so forth. And you really have to be addressing and thinking about both of these in developing these new drugs and evaluating them as well. And the rhetorical question I'll ask you is that will new classes of drugs overcome MDR? And the answer to that question is, well, maybe. Because in fact, sometimes if you're just thinking about MDR referring to just class-based resistance, and you do nothing in terms of permeability or efflux, your new drug or your new class of drugs that is able to overcome a class-based resistance may not make it very much progress if it remains susceptible to permeability and efflux mechanisms. A great example that you may be familiar with have been several efforts to develop drugs for a target known as LPXC and gram-negative bacteria. And several companies have been unable to make real progress in that area. Now, some of that is due to toxicity, but it's actually also due to the fact that you really haven't done much for efflux. And so many of these compounds, even though they have a new mechanism of action and are touted as being some way of addressing MDR mechanisms, they are still highly susceptible to efflux mechanisms. And I think the reason why some of the progress hasn't been made is because, in fact, one hasn't been able to overcome efflux and drive the MICs down low enough so you can give safe doses. So let me take you through then how we're thinking about beta-lactamase inhibitors here. And I want to give some examples here. We're using QPX7728 as an example of how one looks at class-based resistance first and inhibition of beta-lactamases. And what this slide shows you here is the inhibition of nitrocephin hydrolysis. Nitrocephin is a chromogenic cephalosporin that when it's hydrolyzed, it gives you a nice color in your microtiter plate. It's a really easy assay to really quantify then how well you're inhibiting enzymes. This is in the absence of any cells. And you can see 7728 against the important classes A through D there has very potent KIs for inhibition of nitrocephin hydrolysis. And we've included in parallel experiments the spectrum of inhibition that you see with vaborbactam and avibactam, as well as relebactam there. And with notably being that, again, the activity against the class Bs and the class Ds are quite spotty with those other compounds. One can translate that into cell-based experiments here. Again, these are using engineered strains of Pseudomonas aeruginosa with the actual beta-lactamase of interest engineered into those strains. So you have the common background, and you're really able to evaluate then the actual potency of the beta-lactamase inhibitor in combination with various different beta-lactams. And you can see here 7728 works not only with carbapenems, but it also works with the cephalosporins, ceftolozane, and cefepime, but also works with piperacillin as well, as you can see with very, very low MICs to piperacillin there. Although I will point out that it oftentimes depends upon what your partner or companion beta-lactam may be. You can see ceftolozane there because of its rather poor intrinsic activity against the NDM enzyme, even using a beta-lactamase inhibitor really still can't save it from being hydrolyzed by the NDM enzyme. What about mutations then in avibactam? Well, one of the ways that you can do this pretty easily is to look at mutations upon exposure of a KPC-producing Klebsiella to ceftazidime avibactam. You can pretty easily select resistance in the laboratory with this drug. And so these are single passage experiments with these various strains over here on the far left with different backgrounds with KPC, with different companion enzymes there. And you can see then the actual mutants that are actually derived from those passage experiments there involving KPC enzyme mutations. The D179 mutation is the most common mutation that you oftentimes see, but there are also other mutations that can easily be selected in this background. And then you can see then in combination with 7728, in this case here with meropenem, you can see that you can restore the activity of meropenem against these strains. Well, what about other mechanisms now in terms of general or intrinsic mechanisms of resistance? Let's look at efflux first. And so these are strains here with characterized efflux in Pseudomonas aeruginosa with various MEX, AB, EF, or CD, or XY pumps being overexpressed. You can see the non-efflux carbapenem in this case here is biapenem, just so that we can see the contribution of the beta-lactamase inhibitor. And you can see that while vaborbactam obviously doesn't appear to be effluxed by the CD system, you can see that the AB and EF systems do effectively efflux vaborbactam, and hence no activity in Pseudomonas, which many of you know, whereas 7728 is not impacted by those mechanisms. We can then move into Enterobacteriales, or here looking at now both the strains here that will have defects either in ACR-AB upregulation, that's the efflux pump in Klebsiella, or the two key outer membrane porn proteins K35 or K36. And you can see if you want to either downregulates those or makes those non-functional, you can abrogate the activity of vaborbactam against these KPC-producing strains. But then in this case, these resistance mechanisms have little or no effect on the potency of 7728 in that system. So by engineering then the drug to be able to kind of overcome those mechanisms, I think then one moves to the PK/PD questions is then now how do we understand concentration relationships for beta-lactamase inhibitor combinations? And there's a couple of points that I really want to make here that I think are important to bear in mind. One is that beta-lactams and beta-lactamase inhibitor compounds have different dose response curves because they have a different target. A beta-lactam is obviously targeting penicillin binding proteins. Beta-lactamase inhibitors are inhibiting these enzymes. Now, while there are some similarities, they're very different. So you really need to be thinking about these drugs having different dose response curves for each of those activities. And then the goal or the charge to the modeler is to characterize those relationships and then understand how to merge them together as you're trying to develop a dosage regimen for these combinations of each of these drugs. And the dose response curves are going to be different, but they are interdependent. So for example, you could do, certainly if you're just looking at enzyme inhibition, there is no beta-lactam there. There's a dose response curve that will help you understand what the concentration response relationship is for inhibition of the enzyme. Again, when there's no beta-lactam around, because that's how it's working. But if you're looking at then the reduction in the MIC of the partner beta-lactam, well, then that dose response curve that you're going to derive for that beta-lactamase inhibitor is going to be contextualized or really only apply to that partner beta-lactam antibiotic. So if you're looking at the response in terms of what the concentration of a beta-lactamase inhibitor is doing with a partner beta-lactam like ceftolozane, as I showed you earlier with NDM, that dose response curve with ceftolozane is going to look very, very different than with NDM than when you look at ceftolozane, say in combination or in against an ESBL producing strain. And similarly, it's going to look very, very different if you're looking at a very stable compound versus a non-stable compound when you're looking at the same enzyme. So they're different, but they can be correlated depending on what the endpoint is. The second is then, of course, is that you need to be thinking about the dosage regimen of these combinations that consider the dose response curves to the individual agents. And the exhortation here is you don't want to be leaving any beta-lactam activity on the table. Optimize the dosage of the beta-lactam antibiotic, and that's going to be very, very important because that ultimately is what's going to drive the site inhibition of a penicillin binding protein and hence the potency of that combination. So don't leave the beta-lactam dose non-optimized when you're thinking about these combinations. And one way to really think about this, and one way to express it that we published on last year, was thinking about dose-response relationships for inhibitors. Now, for an individual organism, you can certainly look at the beta-lactam MIC that's being tested in combination and the beta-lactamase inhibitor concentration, and you'll get some kind of a typical sigmoid Emax type of curve like this, where with increasing concentrations of the beta-lactamase inhibitor, you're reducing the beta-lactam MIC down to some level that kind of maxes out when all the enzyme is essentially saturated. Well, what happens though? How do you think about this in terms of a population of resistant isolates? Well, we tend to think about, of course, the beta-lactam MIC alone or in combination with the inhibitor. We think about in a population of organisms, the percent they're inhibited, and we oftentimes think about an MIC 90 or 90% of them being covered is oftentimes an important piece, and we think about that in the context of some kind of a breakpoint. Well, for beta-lactamase inhibitor combinations, you might be able to then do an experiment like this against a population of organisms where you've got three different dose response curves with different fixed amounts of the beta-lactamase inhibitor in this setting here. And you would be then looking at, well, what concentration of the inhibitor is the one that you need to bring that population of organisms such that they're 90% of them now are at or below the breakpoint. So you can see in this situation that in this experiment here, what we call the TPC90 or the target potentiation concentration in this case here is going to be a concentration of the BLI potency of 8 micrograms per ml, because we've now brought 90% of those organisms in this example here to the breakpoint for the companion antibiotic corresponding breakpoints. So here's an example. Sorry for the table size here, but I wanted to show how the data look and how one might be able to use TPC90 for doing this. As you can see, here's an experiment where you have a variety of different Enterobacteriaceae here. There's 598 of them, but they're organized according to what enzyme is being produced, because it's very important for us to know of whether or not there is a great difference in a set of clinical isolates in the concentrations that's needed then to bring the corresponding antibiotic to below its PK/PD breakpoint. So across the top here is meropenem alone, or meropenem in combination with up to 16 micrograms per ml of 7728. And what I show here is the TPC90, and that 90% of all the isolates within each of these groups, whether you're talking about a metallo-beta-lactamase producer, which includes NDM, VIM, and IMP, or you're talking about just KPC, you can see then that you have percent of those isolates being inhibited of 90% or better. So that can certainly help us then in understanding then what the critical concentration is going to be for an inhibitor to cover all the enzymes that we care about. Well, then what now as we then start looking at now understanding this potency when we now move into in vivo situation and preclinical models of infection, and particularly in the situation that I've shown you here about, we're thinking about now being able to use this drug with a variety of different beta-lactam antibiotics, but about specific examples. And these are data published about a year ago, looking at 7728 with a variety of different partner or companion beta-lactams here. We have monobactam, aztreonam, as well as the carbapenems, meropenem, and biopenem, and then the cephalosporins, ceftazidime, cefepime, ceftolozane. And you can see this is a strain of Klebsiella pneumoniae, produces the KPC enzyme in addition to some of the other class A enzymes as shown up here. In the in vitro experiments, you can see the MICs all falling down well below the PK/PD breakpoints for each of these drugs. And you can see then that we're also able to translate that activity in vivo. But then, of course, now the next step is to really be able to characterize what's the PK/PD target that's going to apply for each of these drugs. And so to really take you through that, I'm going to switch now to what we had previously accomplished with meropenem vaborbactam, because the work with 7728 is still ongoing. But I'll illustrate the approach here by showing you how it works for meropenem vaborbactam here. So first is going into animal models of infection and probing then the effect of, in this case, humanized exposures of meropenem that are optimized for what you can give in humans, and then looking at various dose and dosing intervals of vaborbactam in that animal model to derive the change in bacterial count over a 24-hour period. And what we found, and you can read about the other indices that were looked at are described in the paper, but the metric that best described the data was the free 24-hour vaborbactam area under the plasma concentration curve, normalized to the respective meropenem vaborbactam MIC. And here are data for 5 strains that were tested within a mouse, neutropenic mouse model, and you can see some of those included meropenem vaborbactam MICs that would be considered to be susceptible, but also resistant, including KP1096, which has an meropenem vaborbactam MIC of 16. But you can see that certainly one is getting 1 to 2 log drops with AUC to MIC ratios here, anywhere in the realm of between 40 and say 90 in this model. We did more extensive studies in hollow fiber models here. These are data from looking at a variety of strains of Enterobacterales in the hollow fiber model, wide range of MICs you can see across the top. And again, the 24-hour AUC to MIC ratio best described the bacterial killing that one sees within this model. And again, in hollow fiber models, you've got a lot more scale. We're using very large inocula, in this case, somewhere around 10 to the 7, 10 to the 8th. And so you can see that you can get a lot more readout in terms of the extent of killing within this model. So then finally, armed with now knowing what the PK/PD index is for the drug and being guided by many of the attributes that I showed you earlier, how do we now put this all together in terms of a dosage regimen in patients? And so some of the key questions that one is going to have to ask about is really understanding the pharmacokinetic properties in humans. And this is an example from Eric Wenzler at UIC that did work for us back in 2015, published in AAC, that characterized the epithelial lining fluid penetration and pharmacokinetics of meropenem and vaborbactam. And you can see here very, very high extent of penetration, as well as actually very similar properties of these two drugs within plasma, as well as epithelial lining fluid. So really understanding that was an important step. And a second step, of course, was understanding now when we're moving into patients, because both of these drugs are largely excreted unchanged in urine, what's the effect of impaired renal function on the clearance of these two drugs? And what you see then on the right-hand figure is the eGFR is calculated by the MDRD equation related to the total clearance of either meropenem or vaborbactam that were measured in actual patients receiving a single dose of the combination. And we were quite struck with how similar these two drugs clearance changed as eGFR decreased. And so this parallel decline in those concentrations was really important, and hence made really dosage adjustment pretty easy here, because a proportional reduction in dose is going to have a similar effect for a given level of eGFR that might be seen in patients. Then, of course, getting real data from real patients is important, and we conducted in association with the folks at the Institute for Clinical Pharmacodynamics, Michael Trang in particular, that a very nice analysis of the population pharmacokinetics of meropenem and vaborbactam in both patients who were enrolled in 2 phase 3 studies, as well as then patients who had been enrolled in the phase 1 programs, not including the ones on the previous slides that I showed you as well. And then really characterize then the pharmacokinetics, population pharmacokinetics, and the variability of meropenem vaborbactam within these patients. And then that became really important for us then to be able to justify a dosage regimen, which turned out to be a 2-gram dose of each of the components administered over a 3-hour infusion every 8 hours for the 2, and then be coming up with recommendations for adjustment of dosage in patients with diminished renal function. And you can see then the data here showing a meropenem vaborbactam MIC distribution for around 1,300 kpc producing isolates, where essentially all of these isolates are inhibited by 8 or less. There's like 1 or 2 that were out here at 16. And then we conducted a simulation of an algorithm that considered both the pharmacokinetics of meropenem and vaborbactam, considered the targets of wanting to have meropenem have a time above MIC of 48%, either getting that MIC either by itself in the case if you even had some KPC that might be susceptible to meropenem, or the joint probability distribution where you have to think about vaborbactam exposures as well, where you would need to be getting vaborbactam AUCs. In this case, this simulation of a ratio of 38, which would be associated with bringing the MIC down into a range by which meropenem could administer it. And this analysis turned out quite nice in showing that we could, for all of these strata of renal impairment shown here, is that we could still get 90% target attainment exceeding 90% for MICs of 8 or less in all of the groups. Now, you would say that the PK/PD breakpoint is clearly justifies 8. That is the breakpoint in Europe, is 8 micrograms per ml. In the US, FDA felt that 4 micrograms was more appropriate because there weren't really many 8s that were treated in the clinical trials of that. But you should be able to look at this and feel very confident that the dosage regimen that's currently approved around the world is going to be able to cover organisms up to 8. So with that, let me just summarize here and state that I believe that beta-lactamase inhibitors are an important strategy for really improving the activity of proven class of drugs. These drugs are beta-lactam antibiotics are arguably the most useful class of drugs that we have for treating gram-negative infections. Having better beta-lactamase inhibitors as well as better beta-lactams, I think is a good strategy for being able to address this problem. And in doing that, you really need to be thinking, as I mentioned before, about class-based resistance as well as these MDR mechanisms. You have to address both if you're going to make progress here. And there are ways to do that, as I've shown you here. And being able to find agents with improved PK and PD properties are going to be important as long as we are able to optimize the doses of these drugs by integrating their PK/PD. So with that, let me just quickly thank those that I've had the pleasure of working with over the years here at QPEX. All the microbiology and biology experiments are led by Olga Lomoskaya, as well as the team that's shown down below that are really exceptional microbiologists and biochemists that have been able to advance this. David Griffith on the non-clinical and clinical pharmacology side, and his team in the pharmacokinetics-pharmacodynamics group doing those animal experiments that I showed you earlier have been important for this. A chemistry group that innovated and discovered these compounds and optimized them as well. The clinical group led by Jeff Loudet that is doing the clinical trials, and then program management, particularly since much of the work that I've been showing you here has been indeed powered by BARDA on a couple of contracts now that have been responsible for bringing these forward, and then the ICBD folks for the really elegant modeling that you saw. So thank you.
Thank you again, Dr. Dudley, for a really, it was very interesting talk, and I learned a lot. You think you know a lot about these drugs, but each time you hear and gain more knowledge, it's nice to hear your story. And I just wanted to remind our audience, please feel free to type in any questions. And I have a first question for you provided by my co-chair, and I'm going to ask you, I really liked your slide about the TPC90, where you showed the activity against a range of pathogens for the enzymes that we are interested. And in your animal model for the thigh infection model, you looked at a relatively high CFU starting inoculum of 10 to the 8th. So I guess, what are your thoughts on the role and importance of the inoculum with regards to the PK/PD index? That's my first question to you. So I just want to give you the opportunity.
Yeah, it's a great question, and I'll sort of answer it. That's the beauty of being able to do work in hollow fiber models. We think it's important to do both the animal model as well as the hollow fiber models. For the hollow fiber models, you can really dial up the inoculum size. If you dial up an inoculum size of 10 to the 8th or even sometimes 10 to the 7th, you'll kill the animal with the overwhelming inoculum. They die of septic shock because you've delivered such a high payload of bacteria. But I think being able to probe large inocula in these models is important. And it's important because the other parameter, which I didn't talk too much about, but certainly is described in some of the papers, is looking for emergence of resistance. And so emergence of resistance can be brought about by small subpopulations of organisms that might have a mutation in a beta-lactamase or might have a mutation in an MDR mechanism, an upregulate those mechanisms. So first of all, if we're going to prove that indeed that all that stuff that I showed you in the earlier, engineering out efflux, engineering out the effect of permeability mutations, and of course not being able to select for mutations in beta-lactamase like you can easily do with vaborbactam, you really need to test all that in a variety of different strains and different backgrounds in these non-clinical models using exposures that you'll be targeting in your studies. And so that's what we're doing now with 7728 is looking at various backgrounds with all these potential upregulations of all of these things to occur in that. Certainly in meropenem vaborbactam, we had, I believe in that paper on the hollow fiber, we studied 18 strains of bacteria. I don't think I've seen 18 strains of bacteria for other drugs that have been out for 10 years that have been challenged in some of those models. So I do think that it's important to look at large inocula if you're really interested in looking at whether your dosage regimen is going to be able to prevent resistance in those bacteria, because we think that that's an important element for durability of these drugs.
No, absolutely. I do agree with you. And I guess as a follow-up to that question, I want to ask you, what about the site of infection, right? So because now within the lung, if you're dosing as opposed to in the thigh or the bloodstream, that poses another layer of challenge. And so what are your thoughts along that, especially with the resistance factor?
Yeah, it's a great question. And so, I think the lung is a good example of where we can get some better ideas by, say, looking at epithelial lining fluid. So the example that I showed there was the biggest question is that we didn't want to get surprised and find out that the drug has some different distribution property into epithelial lining fluid that might present a problem for that. And as you can see there, it seemed to behave in ELF just like it behaved in plasma, where you saw that the pharmacokinetics could be predicted very well from plasma, and that they followed a time curve very similar to that. I think the next obvious question is whether or not you need to be modeling target attainments based upon epithelial lining fluid concentrations. And my own personal view is I haven't been compelled by any data or any analysis that shows that does a better job. And I think sticking with plasma as the compartment and then building the usual link model to what the concentrations might be with an ELF is probably a good way to do that. But I think that what you're really interested in is really understanding then is that you're not being surprised by distribution into those areas, and that you can build the models to really understand whether you have activity there or not. The other special case, of course, is kind of the bizarre things that happen. I'd be remiss in not mentioning, things that happen with drugs like daptomycin or with the older polymyxin drugs where they're completely inactivated in that. Well, then you got a real, that's kind of almost becomes a pharmacodynamic problem.
No, I definitely agree. And I guess with regards to, you talked about dose optimization, right? With the ability to dose the beta-lactamase inhibitor independently with different beta-lactams. And I guess, what are the challenges you foresee with regards to drug approval? The second part is also individualization or optimization problems you could potentially run into, because the testing that has been done with certain combinations and certain pathogens, and then now you're going to change the focus where you say it could become a kind of select what you want. So what are the potential challenges you see with that?
Yeah, I think there's a couple of those. I think, maybe I'll start on the good side is that I think that being able to do a better job of individualizing antibiotics based on their pharmacokinetics is really important. Mark Sheets has done a lot of work on this more recently, talking about the importance of that, even in some cases suggesting that therapeutic drug monitoring may actually be useful. I think that makes a lot of sense. I think we pay a lot of attention to MICs. I wish we paid as much attention to time above MICs or AUCs as we do to MICs, because I think there actually may be more variability in actually the pharmacokinetics than there are in the MICs in many cases. So I think that that's important in terms of patient care, and there are folks like those on the call here that really consider that to be a premium. Secondly, I think that it's not, I think, completely satisfying when you have to start loading up on various beta-lactam, beta-lactamase combinations to get the two drugs that you really want to get together. You can imagine these kind of crazy scenarios where you're loading up patients on ceftolozane tazobactam and ceftazidime-avibactam and perhaps a third one, because all you're trying to do is really access the right two drugs together. And we have other examples in medicine and infectious disease where we use boosters independently. And so a common one that we use is in HIV therapy, that there are boosters that are given independently of the antiretroviral agent to increase the exposures of those drugs. And so I think that, when I look at the future, I think about doing a better job with the pharmacokinetics and being able to really get the doses right of each of these two components together, and perhaps having more information on the diagnostic side. I think, MICs are great, but I actually think that in the future, we're going to do a better job by actually understanding using gene profiling and sequencing to really understand all those mechanisms that I talked about. And I think that combined with the pharmacokinetics are going to actually do a better job for patients.
Thank you. And I think because there is a question in the chat from Sunang where she says, thanks, Dr. Dudley, very informative. What is the possibility of generating a list of criteria for considering the best pair of beta-lactam and beta-lactamase inhibitors for individualized therapy? How far are we? I think the last portion of your answer was kind of directed towards that.
I think that's right. And I think where that's going to go is I think doing a better job now of actually understanding the enzymes and understand the enzyme profiles. So if you have a beta-lactamase inhibitor that doesn't appear to be affected by the MDR mechanisms, the general intrinsic mechanisms, then really the most important thing that you're going to need to know is obviously the enzyme being produced, but then also really characterizing, understanding what the impacts might be on the partner beta-lactam. So for example, we find that for some of these newer strains of Pseudomonas that have ceftolozane resistance mechanisms in them, and some of those are susceptible to carbapenems, but some of those resistance mechanisms seen in ceftolozane are not. You actually find that piperacillin in combination with 7728 is actually the best drug for addressing those resistant organisms. And gosh, you're thinking piperacillin, good grief, that's an old drug. Again, not having to access that by using piptazo plus something else seems to us to be a much more rational way of thinking of things.
Oh, yeah. And I had a question for you. On your acknowledgement slide, you mentioned BARDA, and BARDA played some role. So I just wanted to get your thoughts on how did you find that relationship between a private entity and a government entity, especially given the lack of investment in this area. And you for decades now have been pushing drug development in this area, without much, I guess, funding from big pharma. So what are your thoughts on how did that process help you with this drug development process?
Yeah, that's a great question. So our relationship with BARDA actually started in 2013 around vaborbactam, and it's really interesting. I find some of the comments on those social media and so forth about some of these newer drugs and so forth. It's a little bit kind of reminiscent of thinking about how we've reacted to COVID. A COVID vaccine was worthless 3 years ago. Nobody cared about a COVID vaccine. Well, now we really care about it. And similarly, in 2013, 2015, we were really pretty worried that the whole carbapenem class was on a road to being blown out by carbapenem resistance. Now we've lucked out, and the fact that it hasn't at least spread, and there's been better infection control. So the spread is still there, but not quite as fast as it was when it first came on the scene. But what we did, with BARDA, and the key thing with that is being able to really come up with a program consisting of individuals that really had a lot of experience, being successful. And we could not have discovered and developed vaborbactam in the 8-year period that we had. That's considered warp speed, as you know, for antibiotics. We wouldn't have been able to do that without our partnership with BARDA, and being able to then expand that partnership. I think that we think that the way that we're approaching now our portfolio as a portfolio of products that are covering that end-to-end spectrum, that's a really different type of approach than what other companies and other products have had. And so we certainly, when we talk about commercialization and when we talk with potential partners, they're quite interested in that. Having an IV oral option available that actually gives you continuity on inhibiting a resistance mechanism is pretty attractive for those. And I think we could have a long conversation about where I think the field is going in terms of that. But I think that certainly for products that are really addressing an unmet need, I think they're important. Look, ceftazimab-avibactam is a pretty important drug now. It's not the best drug, I think, to address the problems, but it's an important drug. And commercially, it's now hitting levels that would be considered to be successful for a drug. But we need to start thinking of these drugs, not the way that we used to. We keep wanting to think about them the way we thought about drugs 20 years ago in ceftriaxone. These are kind of like orphan drugs. These are kind of like rare disease drugs. And that all of the way that we prescribe them, monitor them, use them are probably best thought of in that paradigm rather than the old way.
No, that definitely is that's an important consideration. And one very last question, I know, since I don't see any questions in the chat, and I know my co-moderator and me have been wanting to ask you, since you've made the transition from academia to industry, and we have a lot of our trainees on this call, and you've made it both fields work very well and look like, they've both given you a lot of qualities and things that have made you successful. So what I would like to hear your thoughts on that.
Yeah, that's a great question. People ask me a lot of that. And first of all, I say, well, it's actually funny. I don't think of that. There was sort of my career that was in academia, and then there was my career in industry. It's like the old Neil Young comment. It's all one song, and it's all been one, I think, career that really had started in the clinic doing what is now called stewardship and seeing about how one could optimize antibiotic therapy in individual patients, and using the tools that were available in that setting to be able to then to move into a setting and to be worked with other gifted scientists and clinicians to now be able to kind of look on the other end and say, well, I know what it was like to have a drug be released that wasn't very useful. What can I do? What can we do to develop drugs that are going to be useful in the clinic? And I would encourage that what I think a lot about is the next generation of clinical scientists that are going to be involved in drug discovery and drug development. And I think those that are interested on this really shouldn't see it as kind of a fish or fowl, but it's all one song.
Oh, that's a great answer. And thank you so much. And I will let my co-moderator say some closing remarks. Thank you so much.
Sure.
Yeah, there's actually just one more question. If we could just steal a couple more minutes from Gian. He had some trouble with the Q&A box, so I apologize if others had similar issues, but he was asking, since BLIs are sort of like a combination product, why do you think it's challenging to see better efficacy with combinations compared to monotherapy in some of the clinical trials that have gone on? Is PK/PD one of the challenging issues here, or how do you think about clinical trial design and maybe ways that can be improved in the future?
Yeah, that's a great question, and I think I'll start the answer by saying, because you're using two drugs, you've got to really understand two sets of pharmacokinetic indices, two sets of covariates, and you've got to make sure that you've got the dosage regimens for the two drugs to be right. And you can kind of just imagine that if you have one drug, you have a certain probability of hitting target attainment with that one drug. But with combinations, as I implied in that simulation, we had to construct a joint probability that said not only did you have to hit the target for meropenem, but you had to hit the target for the beta-lactamase inhibitor. Now, the good news is that those are all solvable problems that you have. And so, I think that you can, I think you can get as good of efficacy with a combination product, otherwise we wouldn't be doing it, as you can with a single agent therapy product. But I think that, for some of we all know that the trials that have failed oftentimes in phase 3 with antibiotics are because you got the dose wrong. The ribociclib failed in the oral UTI study because the drug had crummy bioavailability, and so you didn't get the exposures that you needed in that. But in general, the beta-lactamase inhibitor compounds have actually performed quite well in the clinical trials where you've been able to do it. Ceftazidime vaborbactam did very well. They never really did a controlled trial against KPC, but certainly in the real-world evidence trials, it certainly showed that it was better than best available therapy involving colistin regimens, whether they were single center studies like those that were conducted by Ryan Shields, or whether they were larger studies such as the Crackle consortium that the NIH did. So I think that I think you have to be careful though, because you certainly could stumble if you don't have the dosage regimens right. And that's why I said, make sure you don't leave any beta-lactam dose on the table. And as you know, there was a lot of use of meropenem at 2 gram doses infused over 3 hours, but nobody had ever done a systematic study of the safety and efficacy of that. And that was a key piece of what we did with vaborbactam. And as we go forward, we think that it's going to be important for other beta-lactams.
All right, I think with that we can close. Thank you, Dr. Dudley, so much for all of your insights and really a fantastic presentation. And I think the Q&A session was also very insightful. So just thanks so much for your time. We really appreciate it.
Great. Thank you. Really enjoyed it.
Thanks, everyone.
Bye-bye. Thank you.